My name is Savannah Conlon. I'm a fourth-year graduate student in Dr. Sheila David's laboratory at UC Davis. I'm excited to share my work with you called the recognition and excision of oxidatively damaged bases in G-Quadruplexes by the DNA glycosylases NEIL1 and NEIL3. My project focuses on a unique alternative DNA structure known as G-Quadruplexes that can form in G-rich DNA sequences. In these structures, guanine bases hexene hydrogen bond with each other, and the layers will stack on each other with the stabilization of central potassium ions. Interestingly, potential G4 forming sequences are found in the promoter regions of many biologically relevant genes, such as oncogenes and tumor suppressor genes. More recently, it has been shown that G4s can affect gene expression. Specifically, the equilibrium between the duplex and G4 structures can turn on or off genes in specific cases. This makes G4s of therapeutic interest. However, because of their high guanine content, G4s are more susceptible to oxidative damage. And when this damage occurs, it can affect the structure of the G4, which could subsequently impact gene expression. Interestingly, the NEIL enzymes can repair oxidative products of guanine, and they can remove these damages from double-strand, single-strand, and G4 contacts. NEIL's best-known substrate is guanidinohydantoin, or GH, as I'm showing here. However, there is very little information known about the repair capabilities of NEIL in G4 settings, and my project aims to determine if various G4 properties will affect removal of GH by NEIL1 and NEIL3. We chose to analyze NEIL's ability to remove GH from 3 different G4 promoters, VEGF, KRAS, and RAD17, in 2 different positions. This is both core and loop damage positions, where core damage is located within the G4 folds and loop damage is outside. To do this experimentally, we take our G4 sequence containing a damaged base, label it with radioactive phosphorus, and fold it in the presence of potassium chloride. We then treat the damaged G4 with NEIL and quench at various time points. By running the reactions on a denaturing gel and determining the ratio of substrate versus product, we can graph the data to get 2 important pieces of information: the rate of excision and the percent GH removal by NEIL from the G4. Generally, we observed the rate of GH removal to be similar for all the G4s, but the striking difference we found was in the percent GH removal. So this is the data of the percent GH removal by NEIL1 and NEIL3, with the sequences being labeled at the bottom, as well as the position of damage, loop, or core. NEIL1 is in dark blue and NEIL3 is in light blue. As you can see right off the bat, NEIL does not excise damage from these G4s to 100%, even though there is excess enzyme present. Additionally, the excision of GH by NEIL is strikingly sequence dependent, with damage in the VEGF G4 being the least preferred. Lastly, we observe that core damage is removed better than loop damage by NEIL. We hypothesize that the sequence dependence of GH excision may be due to varying thermal stabilities of the G4 structure. Additionally, we demonstrate that NEIL can non-productively bind the GH-containing G4s, so differences in repair between sequences could lead to increase in mutations and alter gene regulation. We plan to continue studying these interactions of NEIL with G4s and in the future look into NEIL's role in G4-regulated gene expression. I want to thank you for listening, and I want to express my gratitude to my funding sources, the ARCS Foundation and the NIH, for supporting my research.